and welcome to the present session of respiration. And in this session we are going to discuss about transport of gases. If you just recall, in our previous class we have discussed about breathing or ventilation, as well as we have seen exchange of gases between alveoli and the blood, as well as from between blood and tissues. And now the question is, from alveolar region to the tissue, how does these gases are passed, as well as from tissues, how the carbon dioxide is brought to the lungs. So this is a question, and here we are going to understand that particular process under transport of gases. Now we all know that blood is a vascular tissue, and it is involved in transport of different materials like hormones or different proteins, okay, as well as they also transport nutrients from the digestive tract to all different parts of the body. Similarly, blood is also involved in transport of gases like oxygen and carbon dioxide. So oxygen from the alveoli to the tissue, and carbon dioxide from tissue to alveoli. So don't think that when the blood goes from lungs to the different tissues, it carries only oxygen. It will also have carbon dioxide, but their partial pressures will be different, as we have seen in the previous session. Okay. And oxygen, so that is present in the alveolar blood, should be passed to the tissue, because tissues formed of cells, they need this oxygen for oxidation of the food, so that we can get the energy. Okay, and during this process of cellular respiration, carbon dioxide is produced. This carbon dioxide will be toxic actually if left in the tissue or the cells, so they'll have to be eliminated from there. That's why they'll be brought to the lungs, from which it is thrown out of the body during exhalation, the higher concentration of it. Okay, so first let's see oxygen. Already we know that oxygen is brought from lungs to different tissues, and that blood which has more partial pressure of oxygen is called oxygenated blood, and which is less oxygenated, or which has less partial pressure of oxygen, will be called as deoxygenated blood. And this oxygen from lungs to the tissues is brought through the blood by different means, and one of the method, or one of the mean by which oxygen is brought to the tissue is as physical solution, okay, and then the second one is in combination with the hemoglobin. Okay, either it is brought as a physical solution or as in combination with the hemoglobin. First, as a simple solution, like we are familiar that oxygen can dissolve in water, isn't it? Oxygen can dissolve in water. We have seen it in case of aquatic animals, which can use oxygen only in the dissolved form, and this is taken through the gills. So oxygen is present in the water as a dissolved oxygen, and we are also familiar that the blood is formed of plasma that contains water. So here in this method, or in this way, oxygen will dissolve in the water that is present in the plasma, okay, and it is passed in this process only, I mean in this form only, okay, but the dissolving ability of the oxygen in water is very less. Okay, it doesn't readily solubilize in water. It solubilizes very slowly. So the amount of oxygen that is passed through this method is very, very less. That means, as you can see here, only 0.3 ml of oxygen is passed for every 100 ml of plasma. 0.3 ml of oxygen per 100 ml of plasma, that is the dissolving capacity of the oxygen. So very, very small amount of oxygen, almost 3% of the total oxygen that is present in the blood is transported in this form. Okay, that means this amount of dissolved oxygen in the blood will not be sufficient. Okay, but still around 3% of it will be transported by this method, by direct dissolution or by directly dissolving in the water present in the plasma of the blood. Okay, that is the first way as a simple solution. Now, there is another major method by which oxygen is transported, that is in combination with hemoglobin. Now, hemoglobin, it is the pigment that gives red color to our blood, isn't it? Our blood is red in color due to the pigment hemoglobin. Where it is present? It is present in the red blood corpuscles. Okay, it is present in the red blood corpuscles. That's why our blood corpuscles are red in color. Okay. And this hemoglobin, it is, as the name itself says, it is formed of heme and globin. Heme and globin. Globin is the protein part of the hemoglobin, whereas heme part is formed of iron. Okay, and it is formed of porphyrin rings. Porphyrin ring and iron are present and they form the hemoglobin. And oxygen, when it combines with the hemoglobin, almost 97% of the oxygen that is transported in this method only. Remember, 3% is dissolved in water of the plasma of blood and remaining 97% of the oxygen that is transported will be transported by the help of hemoglobin. When combines with oxygen, it becomes oxyhemoglobin. Okay, and here this combination is possible because heme is formed of, as I said, iron. It is formed of iron. Okay, that forms the heme part of the hemoglobin. As you can see here, this is the hemoglobin heme part of the hemoglobin. Okay, heme part of the hemoglobin. Each one is a porphyrin ring containing iron in it. Okay, iron will be present at the center and formed of porphyrin rings somewhat like this. So that means how many heme parts are present there, units are present, 4 units will be there. The reason is that hemoglobin is formed of 4 polypeptide chains, alpha and beta. 2 alpha and 2 beta chains will be present in hemoglobin as a whole structure. And those alpha and beta chains are nothing but the polypeptide chains formed of amino acid sequences. They fold and each of the polypeptide is provided with one heme part. That means one porphyrin ring with one iron. That means each of the hemoglobin molecule will have how many iron? 4 iron. Okay, those 4 iron will combine with 4 oxygen. Okay, iron of the hemoglobin will combine with 4 oxygen so that each hemoglobin molecule can carry 4 oxygen molecules. Okay, and here I have to clarify certain things. That is, the iron that is present in the hemoglobin is present in the ferrous form, ferrous and ferric form. So it is present in the ferrous form in hemoglobin. When oxygen combines with the hemoglobin, that means when it combines with the ferrous atom, okay, or ion, there will not be any change in the oxidation state of ferrous. There is no change. That means this process of binding of oxygen to ferrous is not oxidation. It is not oxidation because ferrous form will not change to ferric form. Okay, ferrous will not change to ferric. So that means there is no change in oxidation process, state. Okay, that means the process is not oxidation but oxygenation. Oxygenation means a simple adding of oxygen to iron. Okay, simple adding of oxygen. Some weak force of attraction will be present between oxygen and the iron. Okay, it is not oxidation. So there is no change in the oxidation state of the iron. Okay, even though it binds with the oxygen. So that's why the process is called oxygenation. Okay, so hemoglobin will bind to oxygen, which is a reversible process, forming oxyhemoglobin. Oxyhemoglobin. One molecule can bind to 4 such oxygen. Okay, so in this form only the oxygen will be carried to the tissue wherever it is needed. But when it reaches to the tissue, here depending upon the partial pressure, the dissociation of the oxyhemoglobin to oxygen and hemoglobin takes place. Okay, why it happens? See, this binding of oxygen to hemoglobin will be depending upon number of factors. Okay, it will depend on number of factors. One such factor is partial pressure of oxygen. The second factor is affinity of the hemoglobin for oxygen, carbon dioxide and carbon monoxide. Okay, then the temperature, then the protons that are present, that means the hydrogen ion that is present. Okay, so all these which is directly related to the pH of the sample. So based on that, the oxygen and hemoglobin binding will vary. Now, if you consider hemoglobin, this hemoglobin will have high affinity. Okay, this hemoglobin will have high affinity for its affinity towards oxygen and carbon dioxide as well as carbon monoxide. If you consider, it has more affinity towards carbon monoxide. Okay, then its preference is for carbon dioxide and only very less affinity for oxygen. That means if the partial pressure of carbon dioxide is high, then hemoglobin will prefer carbon dioxide over oxygen. Okay, similarly, if there is carbon monoxide in the sample, it will prefer carbon monoxide and not carbon dioxide or oxygen. So we should be very clear. So hemoglobin has high affinity, okay, for carbon monoxide, then to carbon dioxide and then to oxygen. So when this hemoglobin comes to lungs, that is the blood, when the blood is in lungs, we all know that the partial pressure of oxygen is very high compared to carbon dioxide. Carbon monoxide is almost nil, isn't it? So what happens now, because of the partial pressure, which is, we are familiar that it is almost 100 millimeters of mercury. Isn't it? The partial pressure of oxygen in alveoli is 100 millimeters of mercury, whereas that of this carbon dioxide is very, very less. So now what happens? It's 40 millimeters of mercury, okay, 40 to 45 millimeters of mercury. So obviously hemoglobin will have a preference towards oxygen. Okay, it will have more preference towards oxygen. So it binds with the hemoglobin, forming oxyhemoglobin. But when the same thing goes to the tissue, we are familiar that the partial pressure of oxygen in the tissue is less, again 40 millimeters of mercury. Whereas compared to that, the partial pressure of carbon dioxide is 45 millimeters of mercury. It is more than oxygen. So whenever there is carbon dioxide, more carbon dioxide, okay, oxyhemoglobin or the hemoglobin has a affinity towards carbon dioxide, doesn't want oxygen anymore, because the number of carbon dioxide is relatively high. So what happens? Hemoglobin will oxyhemoglobin will dissociate into hemoglobin and oxygen, and now it will prefer carbon dioxide. Okay, it will prefer carbon dioxide, forming carbaminohemoglobin. It will bind to carbon dioxide. So dissociation of oxygen takes place, and this dissociated oxygen will then enter into the tissue, and there the cells will utilize it. Okay, so this is how oxygen is transported from alveoli to the tissue by the help of hemoglobin. Now, based on this information, we can plot a graph. We can plot a graph. As you can see here, we have a graph here, and this graph, where the x-axis will be including the partial pressures of the oxygen, and on y-axis, if you take the oxygen saturation of hemoglobin, the percentage saturation of hemoglobin with oxygen, you get almost a S-shaped curve. A sigmoid curve is obtained, and this curve is called oxygen hemoglobin dissociation curve, and this will help you to understand at what stage of partial pressure or at what partial pressure of oxygen hemoglobin will bind to oxygen, and at what partial pressure it will dissociate. Now you see here, okay, on this axis we are showing the partial pressure of oxygen. Okay, partial pressure of oxygen 20 millimeters, 40, 60, 80 and 100 millimeters of mercury. Whereas on this y-axis we are showing the percentage saturation 20%, 40%, 60, 80 and 100%. Just observe carefully, in lungs where the partial pressure is 100 millimeters of mercury, okay, the percentage saturation of this oxygen is almost 100%. Not exactly 100, but it is almost 100%. Close to 100% rather. Let's say it is 98 to 99% saturated. That means hemoglobin is totally bound with the oxygen. Okay, almost all the hemoglobin will be now in combination with the oxygen. So it is loaded. Hemoglobin is totally loaded with the oxygen, and percentage saturation is up to 100%, very close to 100%. When the partial pressure of the oxygen is 100 millimeters of mercury, okay, 100 mmHg. Now what happens, this blood when it goes to the tissue, we all know that what has happened here you see. So as the saturation partial pressure decreases from 100 to 40, okay, here what has happened, you see dissociation of oxygen from hemoglobin takes place. Dissociation percentage is decreasing, okay, and somewhere at 40, that is in the tissue, okay, how much of oxygen is bound to the hemoglobin? 70% of it. 70% of it. Rest of the oxygen is already dissociated. It is removed. It is dissociated. And if the partial pressure of the oxygen further decreases, more and more dissociation takes place. More and more dissociation takes place. So this oxyhemoglobin dissociation curve shows you the percentage of oxygen binding with the hemoglobin with the respect to the partial pressure of oxygen. Higher the partial pressure of oxygen, more is the oxyhemoglobin formation. Lesser the partial pressure of oxygen, less oxyhemoglobin takes place, formation takes place. That means at lower partial pressure, oxygen will dissociate from oxyhemoglobin, resulting in oxygen and hemoglobin formation. Hope you are clear with this. So on the other side, we are also seeing the ml of oxygen per 100 ml of blood, but I think saturation, percentage saturation is enough. Okay, so here now just if this oxygen hemoglobin dissociation curve can also be used to understand the effect of different other factors. Like I said, oxygen hemoglobin formation will always depend on the amount of carbon dioxide present, the temperature, okay, pH, etc. Now oxygen hemoglobin dissociation curve can be shifted to the right or to the left. Right means even at a higher partial pressure, okay, less oxygen hemoglobin binding takes place. See here, see the partial pressure is almost 120, okay, millimeters of mercury, but the percentage saturation is this point, that means it is less than 90. It is less than 90. Whereas in certain cases what happens, the partial pressure of the oxygen at like 100 itself, it has reached a point, okay, not exactly 100, even before that. You see here, the partial pressure of oxygen is 70, but the percentage of saturation is 90% or even more than that, almost 95%. Okay, so this is 95% of saturation has reached even at a lower partial pressure. Okay, whereas in certain cases, even at a higher partial pressure, the saturation is less than, sorry, that's okay here. Let me use a different color so that it will be easy to differentiate. So here you see at 120 millimeters of mercury, the percentage saturation is this much. Okay, yeah, percentage saturation here. So I can't draw it properly, but I hope you are able to identify that line. Now, the percentage, the partial pressure of oxygen is somewhere here, but it has already reached the peak. It has already reached the peak. Okay, so that means the dissociation curve can be shifted to the left or it can be shifted to the right. And when it is shifted to the left, the left shifting takes place in case of fetal blood, because this fetal blood has more affinity towards oxygen than the adult hemoglobin. So, more and more oxygen will be bound to the hemoglobin at a lower partial pressure. Affinity is high, it likes more oxygen, okay, compared to the hemoglobin present in the adult. Similarly, when the hydrogen ion concentration is less, that means when the sample is alkaline. So, when it happens, usually when the carbon dioxide level is low, okay, proton released will be less. So, that means the sample is or the solution is less acidic or more alkaline. Okay, so in this case also it will be shifted to left in the sense at a lower partial pressure oxygen hemoglobin binding takes place. Similarly, it can be shifted to the right, that means even at a higher partial pressure the binding saturation will be lower to the normal. It happens, okay, when the partial pressure of oxygen is less, one thing. Next, there is a partial pressure of oxygen, carbon dioxide is more. Okay, so this is also known as Bohr's effect. Bohr's effect will help us to understand the binding of carbon dioxide and oxygen to hemoglobin. So, it is depending upon the partial pressure of the carbon dioxide. So, what does Bohr effect states is that at a higher partial pressure of carbon dioxide and higher proton concentration, hydrogen ion concentration, oxygen will dissociate from hemoglobin. So, the oxyhemoglobin will dissociate to oxygen and hemoglobin. Now, what happens? Hemoglobin is available for carbon dioxide binding. So, as at a acidic condition, okay, when the proton concentration is more or when the hydrogen ion concentration is more, what happens? Oxyhemoglobin dissociation takes place. Oxyhemoglobin dissociation takes place. Because of that, hemoglobin will be readily available for binding with the carbon dioxide. That's what Bohr's effect is. And in if you just refer that, it will give you an idea why the shifting of the curve to the right takes place. Why oxygen dissociation from hemoglobin takes place. Now, whenever there is a increase in the carbon dioxide level, hydrogen ion, the concentration will also increase. Okay, hydrogen ion concentration will increase because carbon dioxide will bind with the water, will combine with water forming carbonic acid, which will dissociate in carbonate ion and the hydrogen ion. So, hydrogen ion concentration will be more if carbon dioxide is more. That means the solution is going to become more acidic. So, at a higher acidic state, what happens? Usually dissociation of oxyhemoglobin from oxyhemoglobin to hemoglobin and oxygen takes place. Hemoglobin is anyway formed of protein. So, there will be certain configurational changes. And we already know that there is a high affinity of the hemoglobin towards carbon dioxide. Now, other than that, it might shift to the left even at a higher temperature, at a higher body temperature also oxyhemoglobin dissociation takes place. That means curve is shifted to the right. And one more compound called as diphosphoglycerate, 2,3-diphosphoglycerate in the RBC will also result in shifting of curve to the right. Shifting of the curve to the right means dissociation of oxyhemoglobin to oxygen and hemoglobin. Shifting to the left means oxygen and hemoglobin will bind more and more. Okay, more binding of oxygen and hemoglobin when the curve is shifted to left. When it is shifted to right, it means dissociation is more. Okay. So oxygen hemoglobin curve will help us to understand the dissociation and association of the oxygen and hemoglobin. In other words, it helps us to understand the transport of oxyhemoglobin. Next one is the transport of carbon dioxide. I think we can discuss this transport of carbon dioxide in our next class, but just let me tell you, carbon dioxide transport in blood takes place by three different methods. One is dissolved form, the second one is as bicarbonate, and third one is as bicarbamino compound. By these three different methods, carbon dioxide will be transported in the blood. Okay, so what happens during this process we can discuss in the next class, because we will need more time to discuss each and every step. Thank you for today.